Great, thank you. And thanks so much for the invitation from the organizers. I'm so happy to be here. If I speak very quickly, it's because I've had a lot of coffee this morning, because I landed at 6:15, so I'm still a little bit coffeeed up. So this is great because everything I'm going to talk about has been introduced already, so this will be a perfect, in fact, somebody asked a question after the last talk that is like the ideal introduction to what I'm going to be talking about. I couldn't have planted the question better, because the question was, well, we see that these kinases maybe counteract the action of myotubularin, of MTM1, and maybe trying to balance that could be a potential therapeutic strategy. Well, we've taken that idea and taken it to the next step, which is to look at it in the mouse model, and then also to look at the drugs that may be acting at this pathway to see if they could be potential therapeutics. So I don't have to summarize what MTM is because of Johann's talk, so we're all up to speed there. But one thing I just want to point out again is that this lipid, PI3P, which is the lipid that myotubularin acts upon to remove it, to reduce its levels, is increased in MTM muscle, whether it's from a patient or from any of the models. This is just one of our zebrafish, which I'm not going to show you today, but I'm happy to talk about them later, showing the accumulation of this PI3P. And so we asked the question was whether we could lower this PI3P, and would that provide a potential therapeutic benefit? And this is sort of a schematic of saying that. So in the absence of myotubularin, or MTM1 in myotubular myopathy, you get an accumulation of this PI3P because you have the proteins that make the PI3P still present. So it gets made but not sort of decomposed. And we think that too much of this is what causes the muscle problems. What happens if we now target these kinases to lower the PI3P level? Now, in all cells in the body, this process happens. And there are two sets of kinases, these PI3 kinases, that can make PI3P. And in the muscle, there's two specific proteins, two enzymes that make PI3P. They have very, very unfortunate names. I wish they had easier to remember names like Tom and Joe or something, but unfortunately the names are PIK3C2B and PIK3C3, so kind of a mouthful, but unfortunately we're going to have to work with that, and you're just going to have to remember these little abbreviations throughout the thing. So we're going to be focusing on this one, but both of these enzymes can make the PI3P that the MTM1, the myotubularin, then decomposes or breaks down. So what we started by doing is, because we wanted to work with the mouse model to see if this balancing strategy could work. So we first had to make mice that either lacked this enzyme or this enzyme. I'm going to tell you primarily about this one. So we made mice that basically have none of this PIK3C2B enzyme in any of the cells. We did it both just in muscle but also in the whole animal. This is very dark, but these mice look identical to normal mice, and their muscle is identical. So as far as we can tell, for reasons we don't quite understand, when we get rid of this enzyme, it doesn't have really any effect on the mouse, which actually is a good thing, and I'll point that out in the end. So then we ask the question, given that we know this balance is disrupted in myotubular myopathy, what happens now if in the setting of a myotubular myopathy model, we eliminate this enzyme? Will it balance this out, and what effect will that have? So you've already been introduced to the MTM mouse model. This was provided to us very generously by Anna Bush-Bello, who's here as well, who started the study in 2002, and this has been an incredibly important study and reagent for the field. In our hands, these mice live about 35 days on average, and they have hind limb weakness and get sick very soon, and have all the microscopic features of myotubular myopathy. And they also, as I just mentioned, have increased levels of this PI3P, as we'd expect. The enzyme's missing, so we accumulate this lipid. So here is our strategy. We took animals that had one copy of this and one copy of this and mated them so that we could get three sets of animals. One is the MTM model itself. One is the MTM model itself with just half the level of this PI3C2B. And one is the MTM mouse with no PI3C2B. So the first thing we were able to show is that when we did that, we saw what we were hoping to see, which is that the PI3P level, that lipid that accumulates in MTM and in the MTM1 mouse, goes back to basically normal when we get rid of the kinase. So that makes sense, right? We had just the decomposing enzyme missing. We saw the accumulation. We get rid of the other enzyme that makes it, and it balances back out to roughly normal. So the next question is, does this mean anything for the mice? And we were happy to see that the muscle now looked quite normal when we lose both the MTM and the kinase. And we can show that with quantitation. But even more important, the mice themselves look normal. So this is the MTM knockout mouse, and this is now the double knockout mouse. It's missing both MTM1 and that kinase, the PI3C2B. And so I want to just show you what these animals look like. Belinda has already introduced you to movies of mice, and I'm going to hopefully be able to do the same. I'm going to try to make it a little brighter, if that'll work. So this is a normal mouse. Oh, you don't want to hear me speaking there. This is the MTM mouse at about 35 days of age. So you see it's quite sick and not moving around at this point. And this is now an MTM mouse that also is missing that kinase. So hopefully you can appreciate that this mouse is moving around normally. It looks basically the same as the wild type mice. All right. And importantly then, when we look at the mice and how long they live, it turns out that when we knock out that kinase, the PI3C2B, in the setting of MTM1, the mice are basically completely normal and healthy and live a full lifespan. We now have mice that have lived over 2 years that are missing both the kinase and the MTM1. And so this is quite an exciting result and suggests that when we eliminate this kinase from the very beginning of muscle development, it seems to prevent the phenotype of occurring in MTM1. But of course, this isn't really a clinically feasible strategy, right? For those of you who have children with central nuclear myopathy, you know that usually they're born with symptoms or there's some element of disease that's present at birth. So a therapy, if it's going to work, is going to have to work on an individual who already has some element of muscle weakness. So what we wanted to do next is see what happened if we remove this PI3C2B after the animals are sick, after they've developed symptoms. And we did this using a kind of a tricky strategy where we can knock out the gene at different time points using a drug that induces knockout. And these are the 2 stages we did it. We did it at 21 days of age, which in our hands is when the MTM mice start losing body weight. It's the first time we can really distinguish them from the normal mice at the same age. And we also did it at 30 days where the animals have clear hind limb paralysis. So they're starting to get weak, and we can tell them in terms of their muscle strength being abnormal. We weren't able to do it any later because we were feeding the animals the thing that induced the knockout, and they were no longer feeding very well after that point, so we couldn't really get it done. So this is what it looks like. We were very excited to see that regardless of whether we eliminated the kinase, the PI3C2B, at an early stage of MTM1 in the mouse model, or at a late stage, we got a complete rescue of the phenotype. So we lost one animal right when we started the knockout, but the rest remained healthy, and these animals are now almost a year old. And so regardless of when we remove the PI3C2B, we can not only prevent the disease from occurring, but we can reverse it, which I think is really important from a therapeutic perspective, because it suggests that targeting this enzyme could help even after symptoms have developed. So that's just restated again here. This is sort of all of the accumulated data that we've shown that this works. And again, I think it's just most impressive to see with the video. This is an animal, okay? Let's see, so this animal right here is an MTM knockout at 30 days. So this same animal, we're now gonna feed it tamoxifen to induce the knockout of this kinase, and I'm gonna show you what happens after the animals. So this is now the same animal with 2 of its friends later. So you can see now, it's a little bit dark again, I apologize, but it's moving around again. So it went from not moving very much at all to moving again. So to conclude, when we target this enzyme, this PI3 by itself, we don't see any problems with the muscle. We know that the MTM mouse model has a severe myopathy. When we combine them together, we see basically complete either prevention of the phenotype if we do it before the animals get sick, or a complete rescue of the phenotype after the symptoms develop. And we can therefore conclude that this PI3 is a very attractive target as a potential therapy. Now, a quick side note, because I don't have too much more time, which is that other enzyme, remember I told you that there were two enzymes that make the PI3P? Well, when we look at this other enzyme called PI3C3, when we eliminate this in the setting of the MTM mouse, not only do the animals not get better, they actually get worse. And if we lose just one copy of this PI3C3, the animals are small and they get sick earlier. And then if we lose both copies, the animals are actually never born. They die during embryonic development. This is a little bit of a complication when we think about potential drug therapy. So what are our next steps? So obviously, if we can target this PI3C2B with a chemical inhibitor, this could be a potentially a great possible therapy to consider for MTM1. Unfortunately, right now, there are no pure PI3C2B inhibitors available. So there are any drugs that have been tested in patients before that just target this enzyme. As was mentioned in the last talk, there are actually several inhibitors that can target this enzyme, but also some of the other proteins that make PI3P and other phospholipids that are called PI3 kinase inhibitors. And as I just showed you, unfortunately, if we target this other enzyme, it makes things worse. So one of the issues with the PI3 kinase inhibitors is they target both this and this. So we might not expect them to have much of a positive effect. But we decided to try one anyway. Actually, we tried a whole group of these PI3 kinase inhibitors first in our zebrafish model. And because of time, I'm not going to present them. If people want to find me during one of the breaks, I can show you some cool zebrafish pictures, because we do quite a bit of work in the fish, and it's proven to be a pretty important model for helping understand the disease. But we decided to test one of the drugs, something called wortmannin, that worked in our fish model in the mouse. And so this is showing, first we measured the strength. So we started treating at 21 days, which is when we first again start seeing symptoms in the MTM mouse. And we treated them for, in this case, about 2 weeks and measured their strength. And so here are the normal mice, here are the MTM knockouts, and now these are MTM knockouts treated with wortmannin. And you can see that we restore the strength almost back to normal, basically back to normal with this drug. And now, hopefully, I'm sorry these videos are so dark. So there's two animals here. This guy over here is an untreated MTM mouse model, and this now is a treated one. So hopefully you can see he's running around pretty happy. He's actually going to crawl on top of his little friend there, or I might have stopped it before. But this guy's not moving at all, and this guy has now got movement restored. And importantly, not only do we see an improvement in the strength, in the way that the animals look, but we also see an improvement in animal survival. So almost, we get about a 15-day survival benefit, which it's a bit hard to know what that would mean potentially for a patient. And we don't know whether or not this sort of modest improvement in survival is due to maybe drug levels dropping or the effect on the other enzymes that I mentioned that could actually have a deleterious effect. And we could also show again that the PI3P levels are diminished, but only modestly with this drug. So where are we now? So I think hopefully I've shown you, and you believe, or at least have a suggestion, that when we inhibit genetically this PI3C2B, we can not only prevent disease, but rescue disease in an MTM1 mouse model. And furthermore, when we use a drug, wortmannin, which can act against not only this PI3CD, but also some other enzymes, that can restore muscle strength and prolong lifespan. So what we're doing now is two things. One is we're looking to see if we can develop a specific inhibitor just to PI3C2B. And we're also trying to see if this strategy could help potentially with other centronuclear myopathies. So similar to what Belinda talked about, could this be a therapy that could be used across all the different types of centronuclear myopathies? And given the mechanism we've been postulating and working just on that PI3P, we're not quite sure, but it's certainly worth looking into. So with my last 45 seconds, I do want to make one quick mention, because I don't know if we're going to talk about mestinon at all today. Probably not. So some of you may have heard of this drug. It's also called Pridetostick. And work that I did in collaboration with Anna Bush-Bello a couple of years ago, and has also been done by Francesco Montoni and several other groups, have shown that this drug in models, in zebrafish and in mice, as well as in patients in several case studies, can improve symptoms. And some of you may have tried this medication or have children who have tried this medication. And the big thing we don't know at this point is whether or not it works. So we know it's pretty well tolerated. We know it's used. I know patients that have used it. But what we don't really know is if it's really helped at all. And what we're trying to do is understand whether that's the case. So we could say, yes, this is something patients should be considering, or no, you know what, probably not. And so we've decided to take a survey approach because we know a lot of individuals are already trying it. And what we're hoping to do, we've got it submitted to our institutional review board, and the next month or two is launch a survey, and we'd encourage everyone who's either tried the medicine or have children who've tried the medicine to participate in the survey. Because what we really want to know is, does this drug really do anything for centronuclear myopathy? So that's my quick plug for that, okay? And so I just have to thank the people who did the work. This is my lab. So Nesrin Sabha is the person who did most of the work. Hernan Gonarascu, who I think was at the family meeting in the US, has also contributed. And lastly, I would be remiss if I did not thank profusely the Myotubular Trust, who's been helping support this work. And I think in a really wonderful collaboration in that we had the idea and we've been working with the trust and sort of developing it each step of the way. And that's been really, really helpful. And some other funding sources as well. So thank you. I'll take questions. And that's Toronto.
First question over here.
Thanks, Jim. I wanted to ask you with regards to the Pridetostick study, do you actually establish a neuromuscular transmission defect first on the neurophysiological level, or are you planning to give it to each patient non-regarding if there is a documentable neuromuscular transmission defect or not?
So this is going to be a retrospective survey to solicit the impressions of individuals who've already taken the medicine. My feeling is that there hasn't been a lot of enthusiasm about considering a clinical trial prospectively for Pridetostick, probably because, one, it's pretty well tolerated, and two, as you know, and two, it seems to have maybe some small benefit, but certainly nothing dramatic. So we thought maybe if we could just, it's in some ways kind of a null hypothesis question. If we survey people retrospectively and the majority opinion is that it doesn't really do anything, I think that would be an important piece of information. So really that's been what we've been trying to do. But to answer your specific question, we actually, as part of our natural history study, did nerve conduction studies in RepStim on 18 individuals with MTM and saw no electrodecrement in all 18. None. So I don't know what that means per se. We didn't do single fiber EMG. So basically what Dr. Jongbooth is asking is, have we looked at the diagnostic studies for the nerve-muscle junction in patients with MTM as a way to guide whether or not to try this mestinon therapy? And we actually looked at it and we didn't see a lot of changes. So it's really been based more on just the potential clinical efficacy and the safety of the drug. So.
One more question? Otherwise, I have one, Jim. The Ward-Menin that you were testing, is it an approved drug? And if yes, could you explain what is the advantage of testing approved drugs?
Yes, so Ward-Menin has been used in clinical trials. It's a chemotherapeutic. It's an anti-cancer agent. There's newer iterations of the drug that have been used because there's some bioavailability issues with it. So there's a different formulation of it that seems to be potentially more effective in humans. It hasn't been studied long-term because it's been used as an anti-cancer drug, so the potential safety of it as a long-term therapy is unclear. So I think it's an open question as to whether it would make sense to translate a drug like this, knowing at the outset that there's probably some negative aspects to it, and just in terms of the understanding of the disease, or whether it's worth waiting to develop a better inhibitor to see if that would work. I think it's a very difficult question always to say, when you see a benefit in a mouse model, does it make sense to then, yes, move forward or to try to see a better benefit? And I think if this drug were safer or had better information about it, it would make the decision a little bit easier. And I think Johann's question gets at a broader question of this idea of what's called drug repurposing. So taking a drug that's already been used in humans before and seeing if it might work for a new indication, which is essentially what this would be, though I think this might not be the most attractive drug from that perspective. We have other things that hopefully maybe next year at the Family Conference we'll be able to talk about that are a little bit closer to this kind of repurposing. So,
Thanks. One more question, Nicole.
Is the retrospective study only on MTM or also the other CNMs?
We'd like it for all CNMs because for sure, actually my personal experience is that I think that patients with Dynamin 2 actually respond the best to Mestinon of what I've seen. I mean, I think there might be some mechanistic reasons to support that, but I think it would be interesting to know whether there's any differences or whether it's something that maybe works for one type or not the other. So yes, so all CNM subtypes. Yes. Yeah. Yeah.
I've seen you've used this Warmantine and tamoxifen for the mice. Do you need the same dose as you would need in cancer patients, or is it lower?
So the tamoxifen was used as just as a tool to induce gene knockout. We used a dose that would be relatively comparable to what would be exposed to patients, and so it's actually a pretty safe. The dose we used, the equivalent in humans, has been shown to be very safe. So, but we wouldn't need that strategy in terms of thinking about it in terms of for patients. But does that answer your question, or?
Just wait.
I also wondered about the second drug, this Warmantine. If you think about maybe, you said that the side effects are the problem for using it in humans, right?
Of not tamoxifen.
No, it's the Warmantine.
Of the Wartmanin, yeah.
Right. So side effects are depending on doses, right?
Right.
So I wonder if you need such a high dose to.
Oh, of the Wartmanin. Yeah, that's a good question. So we tried. We used a dose of Wartmanin that's been used by others to study the drug in mice before, and it was picked essentially because it was shown that it would induce certain changes in known pathways that Wartmanin acts on. So we haven't yet asked the question of whether trying different doses might give us a similar effect. But that would be if we could see that a lower dose worked, that might be a better opportunity for therapeutic translation. Yeah.
Great. Thank you very much.
Thank
and
you.
of course, as everybody else, Jim is around for answering more questions. Thank you very much, Jim.
It's yours. Yeah. Thank you.